fungi. So introductory part of fungi we will see at first. Fungi are non-green thallophatic cryptogams. They don't have chlorophyll. That's why they depend on others for food and are called heterotrophs. Whereas chlorophyll lacking organisms call it as a achlorophyllous. So fungi are achlorophyllous heterotrophs. Fungus, it is a Greek word meaning mushroom, and spongus means sponge. Fungi is a large group of organisms, about 1 lakh genera and 250,000 species it includes. The study of fungi, or the branch of botany which deals with study of fungi, is called as a mycology. So mycology is a Greek word. Mycos means mushroom and logos means to study. One can define fungi as achlorophyllous, non-vascular plants reproducing by means of spores and conidia, and possess a typical chitinous wall and true nucleus. We can have different types of molds which causes diseases. For example, mildews, rust, smuts, spots. Also fungi include other mushrooms, puffballs, toadstools, yeast, etc. So, study of fungi, it started from the period of Linnaeus, Carl Linnaeus. Anton D. Barry, a German scientist, is considered as father of mycology and plant pathology. Sir Alexander Fleming, who discovered antibiotic penicillin, is also a dominant scientist who works in the field of fungi. Millardet, the another scientist who discovered the fungicide Bordeaux mixture. Indian mycologists includes E.J. Butler, who is considered as a father of Indian mycology and plant pathology. Then B.B. Munkur is the another one. T.S. Sadasivan, then C.V. Subramaniam. So like these, some of the Indian contributors in the study of fungi. So here we can see in this figure, we can see here the non-mycelial unicellular forms somewhere here. Then pseudomycelium, for example, in case of yeast, So you can see it here. Then these are the unicellular forms, and some other forms are here. So you can see here aseptate mycelium or aseptate hypa, you can say, plural hypi, lump of hypi called it as a mycelium. See here, there are no cross walls in it, and thus the cytoplasm remains continuous, and the structure becomes multicellular. So here in this figure there are cross walls, see, like this, and thus the mycelium becomes septate. Septa may be having pore. If there is pore to the septa, call it as a porate, and if septa is without pore, then call it as a aporate. So here in this figure we have seen some of the forms of fungi. Some more forms we can see over here in this figure. So here plasmodial body in myxomycetes it is found. Then Arceria, so this also belongs to the myxomycetes. This is Stemonitis in the same lower fungi it belong to. This is Rhizopus and this is the Penicillium. So some more forms we have seen here. Some more forms, see in this figure, you can observe some more forms over here. So see here some myxomycetes forms. For example, Stemonitis is here. Then you have Physarum over here. Then there is a Mucor, Rhizopus, then here you can see Saccharomyces, that is yeast. Then here Chitomium. You can see here Aspergillus. Then here you can have Penicillium, which is a Basidiomycetes fungus. Then here Phallus. See here Alternaria. And a Deuteromycetes fungus, Coprinus over here, Coprinus. It is Deuteromycetes fungus. So like these different forms we have tried to observe with the help of figures. Again we will go towards Looking towards the general characters of the fungi. So, as we know, this is a diverse group of chlorophyll-less thallophytes. So, first general feature is related with the habitat. So, fungi are cosmopolitan in habitat, means occur everywhere. They grow as saprophytes, means growing on dead organic matter. They are parasites, means grow on living plants or animals. They may live as a symbionts, for example, lichens or mycorrhizal fungi. Now, habit. Another general character is related with the habit. So, the body, call it as a thallus, growing on or in the substratum. It may grow on the surface of substratum or may be in substratum. Thallus may be plasmodial amoeboid structure as is observed in myxomycetes, and unicellular or mycelial in case of true fungi. Mycelium may be aseptate. In case of lower fungi, this thing happens, or if one can see, for example, in phycomycetes, such aseptate forms are observed, or septate mycelial forms are observed in higher fungi, for example, ascomycetes and basidiomycetes. These are the classes of fungi. Mycelium is composed of large number of thread-like structures called hyphae. It is non-motile. Fungus cell contains, so fungal cell wall is well defined, and it is made up of chitin. Protoplasm is multinucleate. Some fungi are unicellular or maybe multicellular. Higher fungi have tissue organization, and this tissue is called as a plectenchyma. Plectenchyma is formed by branching, interweaving and coiling of hyphae. Plectenchyma may be pseudoparenchyma. In pseudoparenchyma, closely packed hyphae, they are there. Or prosenchyma, where hyphae are loosely woven. Plasmodium is multinucleate naked mass of protoplasm observed in myxomycetes. Fungal nutrition. So, fungi, they lack chlorophyll, hence are heterotrophic. They can't do photosynthesis and thus remains dependent on other or food material. That nature is called as a heterotrophic nature. They may be saprophytic, that is, they take their food from dead organic matter. May be parasitic, take food from the living animal or plant. May be facultative parasite or facultative saprophyte even. Reserve food in fungi is mostly starch or glycogen. Fungal growth pattern. So fungal growth, it mostly takes place at the tip of each hypa. In case of fungal cells, nuclei are small, mycelium may be cynocytic, it means without septa, in which many nuclei are distributed in the cytoplasm. So multinucleate aseptate structure, call it as a cynocytic mycelium. In some other cases, mycelium may be monokaryotic, means having one nucleus in each cell, or dikaryotic, means having two nuclei in each cell. Monokaryotic or dikaryotic in higher fungi. For example, basidiomycetes. In case of basidiomycetes, the mycelium may be monokaryotic or dikaryotic in the particular phase of life cycle. Fungal reproduction. So fungi reproduces vegetatively, asexually, and also sexually. Sexual reproduction is of primitive type. Vegetative reproduction takes place by fragmentation or by fusion or by budding of vegetative cells. Sometimes vegetative hypa breaks into individual cells and they are called as idea. While in some other cases, individual cell becomes thick-walled and such structures they are called as chlamydospores. And from such idea or chlamydospores even, a new fungus gets developed. Asexual reproduction. So it takes place by means of spores in many fungi. In case of phycomycetes, flagellated zoospores are produced. Asexual reproduction in higher fungi like Ascomycetes class or Basidiomycete class and Deuteromycetes take place exogenously, produced conidiospores or conidia. They are responsible for the formation of new fungi. Now sexual reproduction. In fungi, sexual reproduction takes place by union of two compatible nuclei. Sexual union involves plasmogamy, that is fusion of protoplast, karyogamy, that is fusion of male and female nuclei, and meiosis, where zygote divides by reduction division to retain haploid state. Sexual spores produced may be oospores in case of phycomycetes, zoospores in case of zygomycetes, ascospores in case of ascomycetes, or basidiospores in class basidiomycetes. Now life cycle pattern in fungi. It is of three types: sexual cycle, non-sexual cycle, and parasexual cycle. Sexual cycles are characterized by plasmogamy, karyogamy, and meiosis. Non-sexual cycle, in which sexual reproduction is absent. For example, deuteromycetes. And that is why, as there is no sexual reproduction, deuteromycete class is also called as a fungi imperfecta. In parasexual cycle, genetic recombinations occur between genetically distinct hyphae to form diploid nuclei. Mitotic crossing over. No true meiosis occur for haplodization. For example, aspergillus, or you can take another example, that is a penicillium. So where fungi lie, or status of fungi. So fungi are classified with plants even though they have chitinous cell wall and heterotrophic mode of nutrition. So these are the general characters of fungi, and overall survey of different forms of fungi and the general characters related with that we have seen in this part.